Brother Jahan.
Brother Terry, here we are again. Seems like only yesterday.
Completely true. Today we're going to be trying to tackle the ginormous topic of cellular senescence.
It's a big one.
Are we up to the task?
Look, I personally would say no, but there's no reason why we shouldn't try it.
Feeling that imposter syndrome when it comes to these huge topics and trying to break them down in a palatable way for our audience.
Yeah, it's crazy. Crazy. I think to begin with, we could briefly just cover one of the things we've covered before, which is fasting. Because recently I became aware of a hit piece in the New York Times that purported to claim that intermittent fasting doesn't stack up to what we thought it did.
Yeah. There's been a few sort of negative articles on intermittent fasting. This is a big one, obviously, recently published and then made public through the New York Times. There's also a Netflix documentary, one of the going through like health fads and pointing out all the negative side effects. And fasting was mentioned in there because fasting's, its own topic, right? There's its own method and various ways to do it. And there are certainly ways that aren't as beneficial as others. What did you think of the journal article?
Well, I think it fits into this broad category of things that have criticisms of intermittent fasting. And I think you mentioned there are quite a few others. And I think the important thing to point out is that none of them say that intermittent fasting is harmful in any way. Usually what the criticism is, it doesn't work. So what's the point? I might as well be eating all the time. Just, give me a nasogastric and just feed me for 24 hours a day.
Yeah, exactly.
And usually what they show is they'll look at a really specific endpoint like weight loss or something like that, and they'll say like, well, there was no weight loss over this 8-week period of time, and so therefore, let's just, who cares?
Yeah, exactly. So there's a big difference to point out a lack of benefit or less benefit compared to negative effects. I think it's such as in this study, both groups, there was a control group and an intermittent fasting group. Both groups lost weight. That's a thing to point out, which is interesting, right? Because it comes to, if you're part of a study looking at your weight, do you change your habits consciously, subconsciously? There's questions around how they were reporting how much they were eating and what types of food they were eating. They point out themselves that a change in protein intake when you're intermittent fasting could account for some of the losses in lean muscle mass. It's a can of worms, right? Huge.
It's a can of worms. I became aware of this hit piece because I follow on Twitter one of the main researchers in this area, Sachin Panda from the Salk Institute, and he had commented on someone who posted this article saying that, well, studies like this are pretty hard to do actually because compliance is really hard to ensure. One of the ways that he ensures compliance is he has like an app-based method of doing it, and they did something actually quite similar in this study. They had a study app that reminded people of their eating window time, and then they asked them at the end of the study, how was your compliance with the time restricted feeding window? Compliance was pretty good. I think it was like 80% or something like that. Self-reported. But I think it comes down to what we were talking about when we spoke about fasting, was like, you can't really rely on your own estimate of whether you're doing it or not. You really need to track it in some sort of rigorous, objective way.
Yeah, completely agree. Yeah, for a good year, I thought I was hitting 14 to 16 hours, no problem at all. And very quickly learnt that my average would more often than not be around 12 hours, which arguably doesn't hit many of those big benefits that we espouse when we talk about fasting. And you can see other studies who have taken a way more rigorous approach when it comes to compliance, and will have these people in a, standardized environment. Like, you have to come and eat your meals in front of us in this particular building. But as logic suggests that the participant numbers in those studies are really low, because who in their regular day has time to go to a particular place and eat in front of people and do it under strict...
There's a big selection bias there.
Yeah, pretty much so. Guys like you and I, maybe, if we weren't working in the hospital. Yeah, there's a lot of issues there.
Yeah, quite a few. But still, and I think it speaks to this broader issue of you can't really take a study in isolation. One study doesn't mean a lot. The devil is in the detail, and you need to replicate experiments to prove a point or disprove a point. And
like such thing as they're not sexy replication studies, but that's the greasy grime of the work.
It is, yeah. That's the scientific method.
Yep.
But we're not here to talk about fasting, although it is related potentially.
Yes, yeah.
We are here to talk about
senescence. Now, I have prepared a metaphor. So I was a high school teacher before going on to become a doctor, and I'm always trying to think of ways to relate it to particular audiences. And in the absence of tailoring it to someone specific, I've chosen a metaphor that I like, which is cars and racing, like a racetrack.
Yes.
I'm wearing my t-shirt today. So like any metaphor, it's useful to explain some of the more difficult concepts, but there's a huge amount of limitations as well. It doesn't explain everything. You can't take the metaphor, too many steps further down the line, because it will start to not really make sense. But nonetheless, I think it's a very good starting point for those who are a bit naive and don't know much about senescence. But if you do, it might be a little bit laborious, but should be entertaining nonetheless. So I'm
excited.
I'll share the screen. So, in my analogy, we'll be talking about the cell, pictured here with all its machinery inside, as being a car.
Glorious.
Now, cells need to undergo replication. They need to divide and multiply in order to sort of maintain healthy tissue. And it does this through a cell cycle, pictured here. We've got our growth phase in the G1 phase, where we'll grow that machinery you can see inside before we do the big hard job of actually copying the DNA in the S phase. And then we've got a secondary growth phase where we'll continue to grow, where the cells are getting ready to split from 1 into 2, which happens in the M phase under mitosis. Now, in our analogy here, we've got the cell traveling around the cell cycle, which is similar to a car traveling around a racetrack. So we would be starting here on the starting grid, pictured in Albert Park in Melbourne, the Formula One. We'd be starting in the G1 phase. Now, the power of the metaphor allows us to go into this G0 phase. You can see here the cells that cease division. These are cells like cardiac muscle cells, skeletal muscle cells for that matter, nerve cells. These are cells that are functioning well, but don't need to undergo this cell cycle. And you can imagine that as being a functioning car that's sitting in the garage. Nothing wrong with this car, can enter the cell cycle under certain conditions, can go back out on the racetrack, but a lot of the time just sort of stays in the garage, minding its own business. But it looks nice and simple here, the cell cycle, but as a lot of people would be able to understand, there's a lot of complexity when it comes to the cell cycle as well. And the same is true in our metaphor. A racetrack is a lot more complicated than it might seem at first. There's lots of turns, there's lots of danger involved, there's lots of information that needs to be conveyed from drivers to race marshals to the safety people located at each turn. And you need a lot of signals going back and forth to track the cars. And the way they do this in car racing is to use flags. This is a nice, clear way that you can signal from one part of the track to the other, or even to the drivers themselves. And cells do this in their own particular way. In the racing analogy, a green flag means that the race is safe and things can continue. And the cell has its own type of green flag. saying that the cell cycle is safe to continue. And I would like to introduce the term cyclin-dependent kinase, which is CDK for short. It's one of the buzzwords that you'll hear in this area of medical literature. They rely on the CDKs to tell them that it's safe to continue going through this particular cell cycle. Now, you can imagine that on racetracks, some turns are more dangerous than others. Accidents are more common at particular turns. And a cell is very similar in that regard. There are big checkpoints that are pictured here, which is very important that we see a green flag at these particular areas. So there's a G1 checkpoint where we want to check for cell size, the right amount of nutrients are available, or the right amount of fuel is sitting in the fuel tank for the race to continue. There's not much DNA damage, because before you commit all your resources to copying that DNA, you want to make sure what you're about to copy is working well. And similarly, at the end, at the G2 checkpoint, you want to make sure you've had a successful copy, that the DNA that you've copied is worked well, and that the cell is the right amount of size to go from 1 into 2. So there's another growth phase that happens there. Now, very occasionally, things can go wrong along these checkpoints. And when something goes wrong, but it's not major, such as in a race when you might get a puncture whilst you're racing around. You can see here the best car in the world, the Mercedes Formula One car, has got a puncture. And now with their crazy good suspension, they can travel around sometimes even on 3 wheels and get all the way back to pit lane, get a fresh set of tires thrown on and head back out into the race. Now, when something goes wrong in the cell, a lot of the time, if it's not too severe, the cell can fix it itself. And in this analogy, touching on something we've talked about before, I'd liken pit lane to autophagy, where the cell can fix itself by means of sort of cleaning up some of the damage. But pit lane definitely can't fix everything. And when you've got a major accident out on the track, pictured here, you can see that pit lane isn't going to help these cars. You can't get these cars back out onto the track to continue going in the race. And when this happens, a red flag is thrown around in racing. And red flags mean that the race stops. It's no longer safe to continue. And similarly, when different things happen in the cell, such as cyclin-dependent kinase inhibitors, so as the name suggests, we're now inhibiting those green flags from before, the CDKs, or telomere shortening, or DNA damage, too high a presence of reactive oxygen species, or even tumor suppression genes. When these pop up, we get a red flag in the race. It's not safe for this cell to continue to divide. And this broad idea of a red flag being thrown out in the race, or the cell not being able to divide, can be termed senescence. Again, it's not a perfect definition, but it's a good sort of place to start. And senescence is often referred to as the zombie cell. So just to quickly sort of touch on why that is, normally a healthy cell has 2 pathways towards death, which is necrosis or apoptosis, which some of the factors are listed there. Now, senescence doesn't follow necrosis, and it doesn't follow apoptosis. It's a third pathway that leads to this zombie cell. And it's termed zombie because the cell isn't dead, because it hasn't undergone those 2 processes before, but it's certainly not functioning normally. It's not living normally because we've got a red flag out on the track. And the sort of big concept that we will continue to talk about in this podcast is that these cells are actually damaging. And again, using the power of the metaphor, we're talking about cells that are out on track, or cars that are out on track that have undergone damage. And now they're sitting there flaming away, spewing out huge amount of fumes, debris is out on the track. It's certainly not safe for things to continue the way they are. And this has a term in the medical literature of senescence-associated secretory phenotype, or SASP. And we've got extracellular proteases, chemokines, and cytokines that are all pro-inflammatory, that are going to be damaging the cells around them. And as pictured there, as we age, the number of senescent cells increases throughout our body, leading towards this term of inflammaging or inflammaging, where the process of aging is actually causing more inflammation and more sort of damage as we go along. So I'll stop my analogy there before we get too much into student teacher mode. But it's a framework for sort of dealing with senescence, I think.
It's really interesting. This idea of inflammaging, I think, is pretty interesting as well. And actually, you see it clinically a lot. Just by way of example, you often get older patients come in, you just take a routine set of bloods and you've added on a CRP for whatever reason. And even if they don't have an active infection going on, you might notice really like small subtherapeutic upticks in CRP as people either age or are obese or have some underlying condition. It's a really interesting observation you might make in clinical practice, seeing these small increases in CRP. And it's a very non-specific marker, but it's still an interesting observation that matches this idea of inflammaging associated with senescent cells.
So yeah, CRP is just a marker of inflammation or a C-reactive protein produced on dead and dying cells. And if it's going up, and it's can be just related to the mere fact of accepted aging. So, and what's interesting, I found that when you're talking broadly in science, there's correlation and causation. So we can correlate the fact that, and if you have as you age, the number of senescent cells goes up. But then the question is, are they causing the damage? And there's been some really beautiful studies that shows that they actually have. So there's a causal link there as well. So you can take these senescent cells and stick them into mice in particular areas, into the joints, for example, and they'll start to display signs of osteoarthritis. Into the brain, they start to show signs of cognitive defects, which is, we've got good evidence to find that in humans as well. You look into the joint of someone with osteoarthritis and you find a lot of senescent cells. You look into atherosclerotic plaque in and around the heart, and you find senescent cells. You look into patients with Alzheimer's disease, Parkinson's disease, neurodegenerative disorders in general, and there's a high number of senescent cells. Kidney disease, liver disease, all of these things.
One of my favorites that affects men as they age is the prostate. It was probably one of the first organ systems where they really knew that senescent cells were one of the culprits. And it's well known as part of that pathophysiology of benign prostate hyperplasia, where your prostate gets too big and blocks off your urethra as it goes out, and stops older blokes from peeing This BPH pathophysiology is really due to this accumulation of senescent cells causing this microenvironment that allows excess growth in the prostate. So,
yeah, that's a fantastic example that, I mean, a lot of listeners will either have personal experience with, or at least know that their fathers or grandfathers have gone through something very similar. Which leads us to the question, what do you do about it, right?
Yeah, it is a tough one. And I suppose you could broadly classify dealing with senescence in two ways. One is trying to stop senescence from occurring, would be one strategy, and the other is getting rid of senescent cells once they do occur. And so those would probably be the two key strategies from that perspective.
And it's important to point out that, like most things in the body, there's a balance, a seesaw balance of good and bad, right? So senescence isn't always bad, right? We don't want to have zero senescent cells in our body.
It's a really important point. Yeah.
Which is sometimes can be the wrong sort of takeaway message that people can get when you talk about senescent cells.
Yeah. If you think of senescent cells as a trait, that's evolved, and we evolved this process of cellular senescence, then it fits into this paradigm of so-called antagonistic pleiotropy. And this is a fancy term.
Great word, meaning, by the way. Great word.
Yeah, mate, it's a great phrase. It's a phrase that means a trait that benefits you in early life, but as you get older, it affects you negatively. And you can understand that in an evolutionary context, this is beneficial because you want an advantage early on, so that you can pass on your genes, and then the organism literally doesn't care what happens after it passes on its genes. And so then these genes can have negative effects later on. And in some ways too, it's probably a good thing. I'd need to brush up on the literature, but I think the whole cycle of birth and death in a population at a population level is hugely important, because populations need to adapt to the environment they're in. And if organisms never aged and died, then they would never have any incentive to give birth or procreate. They'd never have any, and that evolutionary mechanism to adapt to a changing environment wouldn't exist. And so populations would just die when the environment changed. You know, a new predator came along, and they couldn't exert a selection pressure to change.
Adaptations are that's the clear way to think about it, I reckon, where you need to adapt to the local environment in order to produce better results. That's where I
so in that sense,
really sit.
Aging is such a critical component of that, to avoid parents competing with their children in the environment. They age. So it's a critical component of that.
Because some of the benefits of like regular senescent cells are that SASP that we've talked about, the smoking car sitting on the racetrack, tells you that there's something wrong, right? And so race marshals will run out with their fire extinguishers and put out the car. And in a perfect world, that's what the body does to these senescent cells. It sort of tells the body there's something wrong here, like you've cut yourself and the wound needs to be healed. Senescent cells play a very important role in that process.
Absolutely.
Which is why we don't want zero senescent cells. They have a role to play, but this antagonistic pleiotropy, the role they play positively in early life tends to lead to these huge problems in later life.
Yeah. One of the interesting things that senescent cells do is they play a critical role in embryonic development and early life development.
Yes.
If you don't have the ability to generate senescent cells, or they're just apoptosed, you literally couldn't develop into a normal fetus, into a normal baby. It wouldn't happen. They are critical for regulating stages of growth as well. So one really interesting example is when your growth plates fuse in a growing bone is dependent on the amount of senescent cells in there. This is part of the mechanism why, let's say you're an obese child, their growth plates tend to fuse sooner, partially because they have hormonal mechanisms, sure, but there's also this pro-senescent mechanism associated with obesity. And so if you looked inside an obese child's joint or into the bone, there would be an increased number of senescent cells driving early closure of, or I guess accelerated development and early closure of bones. And so you see this sort of accelerated and stunted development in obese kids.
And
senescent cells play a role.
We've got this chronic sort of inflammation going on in the background, which is largely attributed to the obesity, that sort of too strongly signals to the body that these regular senescent cells that we find in everyone's joint, they're going to get too much attention too early.
It's remarkable. Remarkable. So these cells play a really, really critical role throughout our lifespans, which leads me to this idea that senescent cells, in my opinion, connect what's happening at a senescent level, at a cellular level rather, to the organismal level. A lot of the things we've touched on so far really focus on cellular aging. But for me, like one of the biggest questions you could possibly ask is, how do you connect what's happening at the cellular level, this cellular aging, to the organism? And it's such a difficult question to ask. Because we have billions and billions, trillions of cells in our body, and they're all interacting. You might not necessarily think about it, but a cell in your thigh potentially has some, through signaling in various other ways, has some effect on a cell in your eye. There's a huge organismal metastructure to a body and how cells communicate. And so then the question
flapping
is,
of a butterfly's wings on one side of the world causes a hurricane on the other. The body is a very similar type of metaphor.
It's crazy. And so you have this chaotic system that forms order. And then not only that, it has this metastructure where cellular aging process, there's some clock, right, that occurs at the cellular level that then is translated to the group, to the population, cell population level, which is then translated to the organism level. And so how do you stack these layers of communication as to like the age of an organism? How do you stack that information on top of each other? And I can't help but feel that cellular senescence plays a humongous role in that, where the cell ages, becomes senescent in some way or another. And then you have a small population of cells, and then you accumulate senescent cells, and then that communicates and it sends out a phenotype, sends out signaling, which communicates with the broader organism. And that seems to me to be a key mechanism where you're regulating organismal aging through the level of the cell. If that kind of makes sense.
Yeah, definitely. That sort of zooming out effect, and at different levels that you can talk about different sort of mechanisms or even possible targets to try and treat these things. Right? So a lot of Senolytics, which is a way to say like killing senescent cells, I guess, are targeted at the cellular level, but then there's good merit to say that you should target it at the cellular group level and then the wider organism in general. Yeah, and it's tough. Like when you think about research in general, there are key leaders in particular fields, but they either tend to be focused down on the absolute micro level or the organism as a whole. And there's not too many intermediate steps. Like we as doctors are trained to understand cellular processes, but a lot of the time we're treating big, broad pathologies and just sort of how they present.
Broad brush strokes of like how these these processes work.
Which by itself is a result of the pressure on our health systems and the need to see as many patients as possible to try and affect the greater good. There's a lot of reasons why you can't sort of delve into someone's got this infection right in front of you and you're not jumping into the cellular processes of this particular this particular person. You need that broad brush. There's no doctor that's not busy.
No.
Everyone's under a large amount of pressure. But trying to find the links between those different levels and then finding ways to achieve a good result is an interesting space to sort of exist.
You spoke a little bit about these studies that have been done, essentially giving organisms senescent cells and looking what happens. There are some really elegant studies that really show the pathological consequences of senescent cells in this way, and not only that, the effectiveness of Senolytics. A couple of landmark papers.
Which ones come to mind for you?
There was one paper, I can't remember which lab did it, but they did what you're talking about. They took they took a group of cells, made them senescent, and injected them or like placed them into like the peritoneum of mice, and looked at and saw what happened. And of course, like any good study, it had some rigorous controls. They had these mice that had senescent cells. They had mice that had the same cells but weren't senescent. And then they just had sort of another control group. So you have three controls, or you have one treatment group and two different types of controls. What they showed was that these animals developed a lot of the signs of aging a lot quicker with the senescent cell group. And then what they did was they did the same experiment again, and then gave the animals with senescent cells so-called Senolytics, a type of senolytic therapy, and they improved. They didn't develop the signs of aging that you'd expect, and they were similar to the other two groups.
And similar studies who've done the same sort of methodology have shown absolute regression as well. So we're not only slowing or ceasing the signs of aging, but you can also sort of regenerate that regular, normal, healthy tissue. Which should send alarm bells to everyone listening. Like that's the golden ticket. That's Willy Wonka's ticket to the factory.
But before everyone goes out there trying to find, give me some Senolytics. Unfortunately, it's just not that easy or simple to create a drug that does this. A lot of the best animal models that show these effects are transgenic. And what I mean by that is that you can give these animals a gene that is specific to a gene that you can trigger that will senesce, that will apoptose senescent cells.
Yes.
So let me give you an example. When a senescent cell becomes senescent, certain genes become upregulated, and they produce a specific, and that's what produces our so-called SASP, that secretory phenotype. One such gene I think is called like P16 Inc. something or other. It's like I'm just going to call it the P16 gene. So this specific gene is upregulated in senescent cells, and you can stain for that gene. In one really cool study, what they did was they put another gene. So they made a transgenic mouse where they inserted a gene next to this gene that gets upregulated in senescent cells. And what that transgenic gene is when you give a certain drug, it causes cells that express that gene to senesce. So it's a really elegant way of being able to trigger senescence of apoptose. So when that gene is expressed, you can give the animal a drug, and it causes that cell to apoptose, not senesce.
Yes. One of those death pathways.
Yes, one of the death pathways. And this is really useful because that means that when these senescent cells arise and start expressing this protein, you can give them a drug, and that will kill the senescent cell, cause it to apoptose.
Which is specific, so right? It's that senescent cell.
Super specific.
Presenting that
super specific,
and we can target it. That's like hitting a bullseye
precisely
on the range.
Exactly, which you can't do easily in a wild type animal. You have to have this transgenic model to show it as simply and elegantly and target it specifically. And so in the experiment where they did this and they senesced these cells, man, there were some great results on those mice. They lived 25% longer. Like you're talking about, that cardiovascular disease reverted, their renal disease reverted, everything sort of just got better in these animal models. And that was from really targeted senescent cell apoptosis. Now you can do that, you can sort of approximate that with some medications, but it's not easy. It's not easy at all.
And then you're taking this, you're on the shooting range hitting bullseyes in a nice, calm, controlled environment. And that's what we're talking about here with these transgenic mice. And then compare that to the war zone, where it's really hard to get a bullseye under this crazy, chaotic war zone like area. And that's humans. That's your regular Joe on the street who wants to target anti-aging. Very difficult to find a bullseye in that chaos.
Yeah, there's one particular combination of Senolytics that is currently being tried in a really interesting subgroup of the population. So Dasatinib and Quercetin.
Yes.
So it's a drug combination. Dasatinib is like a monoclonal antibody that has traditionally been used in one of the leukemias. I can't remember which one.
AML I can't remember either.
Either way, it's got a use. Its original use proposition was in a hematological malignancy, blood cancer. But it can be repurposed because it does target a specific senescent cell protein, which helps cause it to apoptose. Quercetin is a herbal derivative, so it's again one of these biological or so-called like herbal supplements that have been refined into a specific molecule. And it's also been shown to target a slightly different pathway and cause senescent cell apoptosis.
And the safety profile of the Quercetin in particular is relatively good, isn't it?
I mean, I think
it's got some drawbacks, but it's a dose-dependent sort of manner.
I guess the safety profile is pretty good. The efficacy is sort of questionable, on its own. As a single agent, it's like, because here's the thing, you might take a medication, orally, it has to get absorbed through the gut, then it has to get into your bloodstream, it has to wheedle its way through normal healthy cells, specifically senescent cells, to cause them to apoptose. And you can imagine in that journey, it's just going to hit a lot of off-target effects, and
Yeah,
you know,
that's
what are the chances it even makes it there without like really whopping doses? I don't know. I mean, it's just it's a kind of, it's a difficult proposition to say, here's a drug, take it and you'll produce your senescent cells.
Yeah.
But that's the dream. And this study, there was one clinical trial done in actual patients using this drug combination, looking at idiopathic pulmonary fibrosis, a really difficult disease to treat.
So got this scarring and stiffening of the lung tissues down at even the alveolar level, the really small level, which causes the lungs to sort of struggle to function properly. Yep.
Totally. And one of the things we know is this fibrosis in the lungs is caused by senescent cells, which is why we use this therapy to trial in these patients. And one of the really interesting findings was, okay, so this was a sort of a preclinical pilot study. So there was no control group.
Yeah.
No control group.
So this is leading into the current study that you've alluded to. Yeah. Yep.
Yeah. So the results of this preclinical no control group study were pretty good. These patients improved in a lot of metrics. And one of the things that is sort of an objective test in these patients is how far you can walk in 6 minutes.
Yeah. And the sit to stand tests as well. And
yeah. And these patients improved. And it's pretty much the first time in this patient cohort that that metric has been able, that they've been able to move the needle on that metric. Usually, you know, trials can improve certain things a little bit, but this therapy actually moved the needle on this metric, which has been historically very hard to do.
Yeah, huge step forward. I mean, obviously, if you're unfortunate enough to have idiopathic pulmonary fibrosis, it's fantastic news for you. But in the broader sense of senescence and treating them, or treating these cells in particular, that's a massive step forward for showing the targeted effectiveness of treating senescent cells or targeting senescent cells. Can't be overstated.
It cannot.
Massive news.
It cannot.
Get excited.
It's very exciting. It's hard to know what the most exciting step forward would be for senescence, to how to target it in the best possible way. Senolytic therapies are good. I know another trial that's happening where they're injecting osteoarthritic knees with a specific other type of senolytic. I think it's got it doesn't have a proper name. It's just got a series of numbers. So I don't know what it's called.
101 or something. I think I've seen
this. It might be that one. It's and so they're sort of targeting osteoarthritis by injecting it in the knee, which is a sort of, I think, probably a better approach, because you're sort of it's a really specific target.
Yeah, and localizing the medication or the intervention to the particular area where we're trying to bypass all that sort of digestive and problems encountered that you've mentioned before. And that's a huge area, right? It's no small feat. Osteoarthritis is the leading cause of chronic pain in the elderly population. We've got some pain medications that show mild sort of improvement, but it's you don't want to be taking pain medication for the rest of your life if you can help it. And actually treating the problem and trying to reverse the problem is a far better approach than just trying to alleviate the symptoms. But it's been traditionally so difficult to do. And this comes back to that time we've talked about it multiple times now, that looking at aging as a disease, right? Rather than just accepting, well, you're getting older, and when you get older, your joints start to hurt. End of story. Rather than saying this is a disease process, our job as doctors is to treat disease processes and, if possible, cure, which is really rare, I would say, actually, in the medical field. Cure is, I'd love to be able to.
I think that's why I say prevention is better than a cure.
Yeah,
it's just there are no cures, really, almost. Definitely. Very rarely. Most medicine is symptom control. Yeah. It's probably a fair thing to say.
And that's why public health medicine that focuses mainly on prevention is traditionally a difficult area to sort of garner interest, because you're trying to make problems disappear from people's view and then asking for more money to make it more effective. And if it's not in the view of our of our governments and politicians, and it's hard to treat. A lot of people have their opinions on this during the current pandemic. We're all staying at home, undergoing lockdown procedures as a way to prevent problems before they happen. I don't want to get too political, but it's upsetting a lot of people to be forced to stay at home. But we're fixing problems before they become big. I'm a fan of it.
One of the things I wanted to say that just to suffix, there not being many cures. I mean, I think antibiotic therapy has been the obvious counterpoint to that, where we
that's a
have
fantastic
been incredibly
point.
good, incredibly good at like curing infective diseases, which is awesome.
That's a really good point. Yeah, antibiotics are very much taken for granted, not only by us as doctors, but by patients, by drug companies. It's hard to sort of keep that ball rolling. There's less and less money in that sphere, but it's a hugely vital, important part of our health system. Definitely.
What's really interesting, I was going to say with antibiotics is what we do with that is the original antibiotic was discovered because you had a yeast that made a penicillin that fought bacteria, and they were just like, oh wow, this thing makes fights other things, because there's a constant microbial war between different microbes. Bacteria have mechanisms to try and beat viruses and vice versa, and we're constantly learning a lot about how this war is waged on a microbial level.
Yeah.
And there are some possible breakthroughs that are happening on the horizon in terms of that, by looking at what these microbes do at their own level and how they defeat each other. So it's a pretty cool area, actually.
It's another shift in perception, right? You go up to a beautiful mountain expanse, you look out at Mother Nature, and it's awe-inspiring. On the first go, isn't this beautiful? Isn't this fantastic? And you zoom in just a little bit more, and you start to see predators. You see lions attacking gazelles and ripping them apart, and bears destroying deers, and it's a lot of pain and chaos. And you go in even further down to the microbe level, and you've got these massive, it's chaos, it's war, and it's battle. But zoom out far enough and things are beautiful.
Taking a cosmic perspective on things is always beautiful.
Yes.
And it's the details that are ugly.
Yes. Yeah, very much so.
But then, by contrast, if you zoom in enough, like, things get beautiful again.
Yeah, that's right.
Yeah.
Devil's in the detail, and so is the beauty.
Look at those pictures of like neurons that are zoomed really close in, and they're just like this amazing, intricate beauty.
Very pleasing to the eye. There's another good example of senescence and how it causes issues. Sarcopenia is the age-related loss of muscle tissue. And in my research leading up for this podcast, there was a really nice example I found of these satellite muscle stem cells. Now, stem cells is a hot topic in biology of aging, and I'm sure you and I will get to it in time. But so you've got normal skeletal tissue, and you've got these satellite muscle stem cells that are just sitting there. They're in quiescent mode. They're the car sitting in the garage. The cell works fine, but it's not doing anything, and it's waiting to sort of enter onto the track, enter into the cell cycle. And when you damage those muscle fibers through, deliberately working out in the gym or through a major injury, these satellite cells are activated, and they'll migrate to the area. They'll enter the cell cycle. The car will go out on the racetrack and start to multiply, proliferate, and sort of repair that tissue. Now, with aging, we see these satellite cells that are sort of sitting in the garage waiting for their call to the track. They start to senesce. So the car breaks down while it's sitting in the garage with aging. The engine starts to get a little bit, sort of clogged up. The wheels are degrading in the various temperatures and everything like that. That's just sitting there in the garage. So then when you have this muscle damage, the satellite cell can't activate, go to the area and start proliferating. It's a senescent cell. Now it's just sitting there as a zombie, not knowing what to do. And they've been able to, in vitro, I think, so in the lab, they've been able to cause these senescent cells to reactivate back into their muscle stem cell. And then, boom, they're ready to go. And they can go to work. And so for fit, healthy guys like us who want to maintain our healthy muscle tissue as we age, that's a really exciting endeavor. I mean, look at your
It's super
workout
exciting.
equipment behind you. You're a healthy guy.
Yeah. Allegedly, yeah. So you mentioned these satellite cells, really interesting. So every tissue that we look at, almost all of them have these sort of stem cell niches. And what a stem cell niche is essentially like a little pocket in the tissue where there are slightly undifferentiated versions of the differentiated cell types in that tissue. So in your example, you're talking about muscle cells. These satellite cells can then become muscle cells and replenish the population. So satellite cells form part of this muscle stem cell niche that helps replenish tissues in a normal way. And as you mentioned, as we age, these stem cell niches across all tissue types actually degrade.
Yeah.
And part of the mechanism of that is probably senescence and the SASP as well, in that local tissue microenvironment signaling, causing tissue degradation in a specific way by virtue of the SASP expression of these senescent cells. That's been shown in the gut, in the gut stem cell niches, which are hugely important because the gut has huge cell turnover. You know, the inside, the mucosal lining of the gut is constantly having to be replenished and replaced, a bit like our skin. It's a very similar sort of surface, and it's constantly shedding, constantly needing to be replenished. So these stem cell niches are hugely important in the gut, and as we age, they get worse, and and senescent cells are certainly partly responsible for that. So Senolytics might help the stem cell niche recover to some degree and allow you to replenish healthy tissue in part, and that might be part of the process of what's happening. When we use Senolytics, we'll get rid of senescent cells. Pretty interesting.
That's such an exciting area. I love the topic of senescence because you can link these problematic cells with, if we can just attack them in a particular way, then the idea of stopping aging is obviously appealing, but the idea of reversing aging is phenomenal. I mean, the body has endless ways to demonstrate that it's really good at fixing itself. You know, just the idea that if you cut yourself and a few weeks later you can't even tell that there's anything there, and if we can optimize conditions where we can do that through all aspects of our body, then it is perceivable that you stay fit, young, and healthy. And if that doesn't make you excited, then I don't know what will. That's future needs to be exciting, and this is the future.
It's pretty exciting. Yeah,
definitely.
One important point to mention as well, I think, is senescence is one of the mechanisms by which you can avoid cancer cells progressing. So senescence is part of this cell cycle checkpoint, and traditionally, if you know your telomeres became too short or there was too much DNA damage, then you would either force down into senescence through either the G1 checkpoint or the G2 checkpoint, depending on what type of damage there was, whether it was telomere driven or not. And so one of the concerns with getting rid of senescent cells was this idea of would it promote cancer growth by the fact that all of a sudden there isn't these senescent cells taking up space and you're getting proliferation of cells more than you normally would. And interestingly, in these mice experiments where they get rid of senescent cells, you don't see an increased incidence of cancer. In fact, you see less cancer. So
that's an interesting. I'm glad you touched on that. It's an important point to sort of put forward. That's one of the beneficial ideas of senescence, is that if your body picks up that you've got a cancerous cell through this DNA damage or various other ways, then you want to arrest that cell. You don't want it to proliferate. You don't want this cancer to get bigger and bigger and bigger. So you want it to enter into that zombie state. And then ideally you want to have your immune system come on and kill that cell. And that's another avenue, I guess, in this sphere of senescence, the idea that we can just sort of give our own immune system a kick in the butt to go around and find these senescent cells and clear them. That's a whole nother can of worms. While I remember it, you mentioned telomere shortening, and I've got a nice shoelace to Terry coming out as well. So you can think of a shoelace being our DNA, our DNA strands, a double-stranded helix.
Could I just is that from, where's that shoelace from?
It's from one of my military boots.
I know, it's I thought it would have been, it looks super military colored.
Yeah, it's one of the boots. I don't wear that often right now, so I figured I'd... and you've got these little caps that most people are familiar with on the end of a shoelace. Now, why is that there? That's there because if you didn't have it, it would start to fray. The shoelace could degrade over time. It's hitting the ground, all sorts of different damage. Your DNA has shoelace caps. It has things sitting on the end that help protect the DNA. And you can think of this little cap as being a telomere. Now, with time, every time you double the DNA, a little chunk is sort of taken out and it gets shorter and shorter and shorter and shorter. And then when it gets too short, the body says, we don't really want to copy this shoelace anymore. We don't want to copy that DNA because there's issues for how long it can stay around, and we might screw it up when we do copy it. So you senesce those cells. They enter into that cell cycle arrest. Red flag goes out on the racetrack, and they get put off to the side.
But that doesn't happen in our stem cells.
Yes. Yeah.
So in areas where we make our gametes, like things like sperm and eggs, in areas where there are stem cells, like in our bone marrow and stem cell niches, there's a special enzyme activated called telomerase, and that helps rebuild those telomeres.
Yeah, which you mentioned in our previous podcast, the lobster has
almost ubiquitously activated telomerase everywhere. There's
not just the stem cells. A few little things.
And I think it's really important to note that even though these creatures are, in a sense, cellularly, biologically immortal almost, or that's what it would appear, they still do have this evolved lifespan, even though it's through a different mechanism, which shows you how important it is. A European lobster can live like 30, 40 years or something, but it's still going to die from molting and from the energy expenditure of that. So it's just done it through a different mechanism.
So it's one aspect that we want to sort of keep in mind and target if possible, make interventions through, but it's certainly not the whole picture when we're talking about combating aging, is it?
No.
Which is the job of the podcast, right? We want to point out, I think telomere length and shortening and issues with telomeres is garnering a lot of sort of public interest. I wouldn't say most people, but a lot of people would sort of know the idea of what you're talking about there. But we want to help expand the knowledge base.
There was a really interesting controversy that occurred a little while ago. I can't remember how many years ago now, but it concerned lab mice and telomeres, and a problem with lab mice and telomeres. And this problem was discovered by Bret Weinstein, who people might know from
the Joe Rogan podcast. Yep.
Yeah, I should actually say Weinstein. It's Weinstein. From he's made a few appearances on the Rogan podcast. He's got a great podcast himself. So does his brother. But he is a biologist, an evolutionary biologist, and he noted this problem in lab rats, essentially, that they had really long telomeres, excuse me, compared to their wild type colleagues. And this was a problem because they had literally bred this mouse model to have long telomeres and the result of that is that cells would tend to become cancerous more often, and so you had a mouse model that developed cancers a lot more easily. And so when you were and developed cellular senescence less frequently, so you had a mouse model that tended to exaggerate the cancer risks of drugs and various other things, and was not really a correct model for testing the side effects and risks of drugs.
And this was almost universally used in the scientific, right? These particular breeds of mice, and they've been sort of bred up and multiplied and sent out to various areas. But that whole family can be somewhat thought of as being tainted, right? And that's a
little bit, for that particular use case, I guess. For other use cases, doesn't really matter. But pretty interesting.
Yeah, very much
Interesting.
topical for our discussion.
So yeah, telomeres. I see them as like being a part of the picture, but they're certainly not like the whole kit and caboodle.
Yeah, an important part, but not. It's the wheels on the car, but there's still the engine involved. There's still the track conditions. There's still the aerodynamics. Yeah, it's
because I mean, there's a lot of ways a cell can become senescent. Telomere shortening is just one of them, and there's a lot of different ways a cell can become cancerous. One of the key things about a cancer cell is that it activates telomerase, and so it can become biologically immortal.
Yep.
But there are actually certain cancer subtypes who have really short telomeres for whatever reason. I can't remember what they are now, but they exist. And so there's always there are these sort of rules, telomerase shorten, telomeres shorten, cause senescence, but then there are things that break those rules.
Yeah. And that's part of the issue, I think, when you switch between a medical conversation amongst colleagues to talking directly to patients. It's not altogether appreciated, those subtle differences. Cancer tends to be grouped: anti-cancer drugs or anti-cancer practices. Fasting's good for anti-cancer, but it's a world unto itself, right? With all its devilish little details, and depending on which cancer and what sort of circumstances. And it's not like I have the solution there. It's just it seems to be a by and by fact of life that you're constantly having to reframe perceptions and expectations about medical treatments, tailoring it to this particular patient. And sometimes it's sad, that you're dealing with a patient who's got a preconceived idea about how they're going to get better, and it's really it's almost the complete opposite way to how you're trying to direct that patient in the way they need to go. I wish it was as simple as people want it to be, but that's why medicine is hard, right?
Yeah, it's constantly just really complex multivariate problems that we don't actually understand very well, and then trying to find an adequate solution. So yeah, definitely, it can be pretty frustrating.
Well, I think we've made a good headway into an introduction to cellular senescence and senescence for the organism in general. As people can probably appreciate, we didn't get too far, and that's because the topic is so broad and interesting. There's so many rabbit holes for us to go down to. So this isn't the end of senescence. You'll hear Mission Methuselah coming back around on this topic and incorporating it in many ways. But
definitely.
All right.
Didn't even touch on caffeine at all.
Oh yeah, actually, let's just finish off with that the turmeric and caffeine that we were discussing before the podcast.
So just for a little ray of sunshine, something simple that maybe you could do if you're a coffee lover and can tolerate caffeine. There was a pretty interesting study came out February this year that was that looked at the effect of caffeine on senescent cancer cells. So to explain that a little bit more broadly, when you treat cancer with chemotherapy, one of the things that can happen is that the cancer cells actually become senescent instead of dying. That's still good because the cancer is suppressed, but unfortunately, it does mean that there's a risk later down the track that this senescent cancer cell can unsenescent.
No longer growing and proliferating. It is still sitting on the track, smoking away. Not great.
And the annoying thing, the annoying thing too is that a senescent cancer cell isn't going to die as well from chemotherapy, because oftentimes chemotherapy is targeting highly proliferating cells, cells that are dividing a lot. So they tested a bunch of compounds to see which ones would kill these senescent cells, and they looked at turmeric, they looked at curcumin, I should say, one of the compounds in turmeric. They looked at caffeine, and there was one other one they looked at actually, but I can't remember what it was.
I'll look it up while you're talking.
So they looked at these compounds to see if they would cause these cells to senesce, and caffeine really, in a cool, and curcumin didn't really help, I believe, but caffeine and the other one did.
In a very big way.
And yeah, in a pretty reasonable way. And so just to explain that a little bit more, these were cellular models. So they were growing cancer cells in a petri dish, treating them and looking and seeing what happens. But what's really interesting about this study is it sort of matches some of the epidemiological evidence that's come out surrounding coffee consumption in cancer.
Yes.
So there was a really interesting study that looked at a large cohort of, I can't remember how many now, but a reasonably sized cohort of metastatic colorectal cancer patients.
The cancer of the bowel that's spreading around the body.
Correct. And they found that people who consumed more coffee tended to live longer. Interesting.
And this is a possible mechanistic way to explain that, right? The other
totally.
The other intervention was thymoquinone.
Everyone loves a bit of thymoquinone for sure.
Yeah, who doesn't, right?
Yeah.
Yeah. But the idea of turmeric coffee, perhaps holds more weight. Although, as we've pointed out, the turmeric wasn't the big key player, but caffeine. I mean, who needs more excuses to drink coffee?
Sweet cup of joe.
That's it. Most addictive substance, but I love it. I'm a huge coffee fan, and this will just be one of the things in the arsenal where I'll use it as a rationale for my heavy consumption.
And look, there's some evidence that coffee can help with cellular apoptosis, or can cause cellular apoptosis, which might help this whole senescent apoptosis picture. I would say it's not going to be a big effect, but like it's probably got some. It's not zero.
So. Yeah. And it's a leap to link caffeine, senescence, and Alzheimer's disease, but there have been other studies that show that caffeine consumption and Alzheimer's disease are linked, and the more caffeine you produce, the less your chance of Alzheimer's dementia. And I mean, perhaps there's a senescent link there as well. We've definitely correlated senescence and Alzheimer's disease, and and correlated caffeine and Alzheimer's disease, and there could be a mechanistic path there as well.
Absolutely. Watch this space.
Yes.
And drink your coffee, maybe.
That's it. All right. Well, thanks for your time, mate. Let's we'll reconvene for the next episode. But that's senescence as an introduction. All right.
Absolutely. Yep. Hey guys.
This is our end of podcast medical disclaimer.
Yep. Although we are medical professionals, this is not medical advice, and this does not create a doctor-patient relationship.
It's not prescriptive and not intended to be so. We always recommend seeing your primary physician in the first instance. This is mainly for education and entertainment purposes.
Absolutely.